My friend.
Go get them.
All right. It is an honor and a privilege to be here in this great room of minds who are delivering the next generation of healthcare innovations to improve the lives of patients. As Matt mentioned, my name is Rome Madison, host of the Genetics for Healthcare podcast. It's a podcast for patients where we share patient stories and we talk to experts to talk about the implications of our genetic and genomic signatures for our healthcare and also when we're battling diseases. I want to, from the beginning, be intentional because everybody came here for a reason. I was just sitting here talking to my friend Stephanie. I'm bad with names, but I'm good at short term. But we were just talking about why we came here, and she talked about the variety of thought, experiences, and things like that. So I want you to take a minute at the top of this conference with the paper in front of you or wherever you're taking notes, set an intention. You came here for something, whether it's information, resources, opportunities, relationships. I want you to be intentional about making sure you get what you came for. And I'm sure there's a lot of reasons why you came, but I want you to take a minute and write it down and be intentional. And while you're doing that, a little background. I've been in this industry that we call precision medicine for more than 2 decades. In fact, I started with the first company the same year the Human Genome Project completed. And at that time, it was a fresh tissue assay that required us to kind of put together this omni-channel strategy in order to get one report for a patient. Now, you think it's hard to order a test because we have liquid biopsy, archived tissue, and so many patients aren't getting these tests to have access to your therapies and clinical trials. Imagine that, right? This required fresh tissue, and this was the early days of precision medicine. It was the first assay that identified patients who were the most and least likely to respond to therapy. At the time, chemotherapy was the hallmark of therapy. There was only one targeted therapy, and that was Herceptin. But as years went on, more targeted therapies came and we added those biomarkers or FISH tests to this package of information so that medical oncologists can customize, be more precise with their therapy, their therapeutic approach for these patients. And so to get a report, we'd have to start with the medical oncologist to go over the data and show them why this information is relevant, give them a value proposition. And even if they said, you know what, I'll try it. We then had to go to the hospital because the hospital had to send out fresh tissue. These were chemoresistance assays to identify what's not likely to work for those patients so we can save them from excessive toxicity, the unnecessary cost, and save time with trying to find something that will work to make the first shot be the best shot. We're still asking the same questions today, even though the technology has advanced quite a bit. But once pathology said, yeah, we'll work with you, some of them didn't, for whatever reason, they didn't want to get involved. But once pathology said they would work with us, we then had to go to the surgeon because the surgeon knew the best chance for this patient to have long-term survival and a quality of life is if you can completely resect the tumor. And then we had to go back to pathology because once the surgeon had a patient in the operating room and they're doing a mastectomy, they're doing a colectomy, they're doing a lobectomy, a craniotomy. They had to get that tissue out and make sure they give an order for the test because downstream, that tissue had to be fresh. It couldn't be put in preservative, in formalin, because we'd missed the opportunity to get these genetic and genomic insights for these patients. And if it's all successful, then it makes it out of the hospital and then a report gets back to an oncologist. That's hard, right? That's hard. But here's the thing. Over that period of time, now this is 10 years before NGS was a commercial technology. So how were we able to get people on board with this terrible workflow? Because one communication gap, one communication error, the lack of communication, the whole thing is destroyed, and the oncologist and all the stakeholders that want this information for a patient can lose interest. The way we were able to do that is by putting the patient first in the minds of everybody who would touch a patient specimen. I would not hesitate to tell a clinician that a foundational principle of the Hippocratic Oath that you took was to do no harm. I would not mind telling them, because I knew that they studied in their fellowship or in their residency, principles and practices of oncology with Dr. Vincent DeVita, who said the primary reason chemotherapy or systemic therapy fails is drug resistance. That's the heterogenetic nature of cancer. With drug pressure, mutations happen, it figures out a way to survive. And we also had to talk to everybody in the operating room, the circulators, the nurses, the frozen section staff, the histology department that would get that tissue and properly harvest those cells to send it out. And we would tell them, if you're fortunate enough to live for a long time on this earth, chances are cancer will impact your life. Because according to the American Cancer Society, 1 out of 2 men, 1 out of 3 women will be diagnosed with cancer in their lifetime. And if and when that happens to you, God forbid, someone that's close to you, a loved one, a friend, or even a casual acquaintance like a neighbor or a coworker, would you want to be exposed to chemotherapy that, if we look across all tumors at that time, was effective in 2 out of 10 patients? Or would you want them to look at your unique biological signatures, get your genetic and genomic insights, and customize the therapy to your signatures? Of course, they would remember that when they saw a patient and the order came to take the proper care and process to get that tissue out of the hospital. Now, you would think that that would be enough for people to say, you know what, I'll try it. When is more information a bad thing? But that's not the case. Over that period of time of going and trying to get people on board for a personalized approach, I spent over 20,000 hours in the operating room watching tumors be resected from every part of the body. And early in that time, a mentor of mine told me that the path to adoption for precision medicine would follow what German philosopher Arthur Schopenhauer called the 3 stages of truth. The 3 stages of truth is that it's first mocked and ridiculed. You have no idea how many doctors I sat in front of having this conversation around biomarkers, the value of getting personalized genetic and genomic insights. And they would sit there and go, biomarkers? Biomarkers have been around since the 70s. If they were any good and had any data around improving patient care, we'd be using them routinely already. I sat down across from a clinician. He listened to my entire presentation. He got up and told me, "You're selling snake oil," and walked out. Even the surgeons who would actually work with me as they resected tumors the size of footballs, y'all not hearing me. They said, you know what? I admire your effort. I'm going to give you as much tumor as you want to test this patient. I don't know if they're going to use the report, but I'm going to support you because you got a hard job, buddy. You see, when new information and new ideas come out, some people who are considered experts or who are comfortable doing the things that the way it's done, it's so new to them that they respond with laughter and just dismiss it. But eventually, they move to the second stage of truth, and that is violent opposition. When new ideas start to take root and they gain traction, it threatens the established norms and long-held beliefs. I'll never forget, I was at an academic center, and I was talking to a breast oncologist. And we had a good conversation, and he was supportive, but he was still a little hesitant. And at the end of our conversation, he says, you know what? I hear what you're saying, but I can't even think of one time where this was requested by a family member or a patient. I said, oh, tell me about it. What was the situation? And he looks and he has this look on his face like, hey, it was a doctor friend of mine's mother, and their patient wanted this information. As I waited for the words that came out of his mouth to take root in his own head? I said, I said, you know what? I can understand where they're coming from, because if my mother had breast cancer, I would want this test information. By the way, doc, you're a breast oncologist. Those rooms are filled with patients. Chances are those women are mothers. And so when you see a mother, please order the test. How y'all think that ended? Violent opposition. I was not welcome back. But eventually, we moved to the third stage of truth, which is it's accepted as self-evident. When new information comes out, it becomes more robust, and it has the potential to change practice or societal thinking about the way things are done change. After 2 years in this business of going around getting laughed at, getting thrown out of hospitals, because I didn't have anything to lose, really wondering if this is something that people wanted, I arrived at this third stage of truth when my own father was diagnosed with colon cancer. But fortunately, the hospital where my dad was treated and the surgeon who did his colon resection were my clients. And so when the surgeon made the connection that this is my father, he called me on the cell phone the day before the surgery and said, well, Rome, do you want to come to the surgery to make sure your father gets this test? Of course. I was able to sit or stand tableside as the colorectal surgeon combed through my father's large intestine and found the golf ball-sized tumor in the ascending colon. I stood tableside, watching him comb through every lymph node in my father's bowel, examining them for infiltrating tumor cells. He did an intraoperative ultrasound looking for metastatic lesions in his liver and examined the peritoneal cavity for tumor implants. And even though we had surgical masks on, I knew from the look in his eyes that he was smiling when he turned to me and said, we got it all. Because he showed me the clean margins on the bowel when he stitched them back together. Fortunately, my father had a favorable molecular profile. He responded well to chemotherapy, and today he's a 20-year colon cancer survivor. But here's the thing. What if I gave up? What if I let people's mockery and laughter deter me from thinking that this is even important or people wanted it? What if I had not put in the work to get that hospital and that doctor on board? What if I didn't know? My father would be one of these statistics that we see every single year. 20 years ago, we were asking the same questions. How do we identify patients that are most likely or least likely to benefit from therapies? And then even more so, how do we get access? How do we get the patients to get access to these tests and these treatments? Even though the technology has improved many-fold, we still have disparities. Pancreatic cancer, less than 15 of eligible patients are receiving germline testing, despite a 100% for everybody recommendation with the NCCN. This past year, or 2005, last year, doctors are saying the reason why they don't order these tests that can unlock access to these new therapies that you're bringing to the market, these clinical trials that you need to recruit patients for, insurance denials, even payers haven't gotten to that third stage of truth yet. But you know how we're going to be able to do it here together? It's by putting the patient first in everybody's mind that we come in contact with. With every team call, every sales meeting, every time you speak to your IT department, your market access team, your managed care team, your training department. How can we better enable them to put the patient first in everything that we do? I submit for you to consider, for is all the work that's going on here that are bringing new therapies, new clinical trials, new diagnostic tests, new insights to genomic and genetic insights in order to improve patient care. None of it means anything if patients can't get it. So let's be intentional from here going forward. That's my QR code. We're here for collaboration. If you would, scan that. Because what I'm excited about my platform is I get to work with patients. I truly believe that disruption and the growth of precision medicine and new innovations are going to come from patients. We tell these patient stories so loved ones can know when they're in that situations, the language and the confidence to have these conversations with their care providers and their family members. But I also get to work with industry, pharma, diagnostic companies, surgical companies to better enable your leaders and your sales team. Because with that bottom up and top down pressure, that's how we're going to close the gap between innovation and patient access to these marvelous, wonderful innovations that all of you and your teams are bringing to the market. But if we don't collaborate, if we're not intentional, the same thing is going to keep on happening year after year after year. We're going to be talking about how many patients don't have access to this. Is this good or is this good? Is this helping somebody? Y'all can talk back to me, right? Y'all not dead. Y'all not asleep out there, are you? Is this good for somebody? Is this helping somebody just besides me? All right, that's good. Okay, so as Michael Buffer said, all right, let's get ready to rumble because coming to the stage is a